So hello. Yeah. So I was asking, what did we do last week? Can you guys recall about it? Yes. This is week three, right? So during week two, what did we do? Like we majorly discussed about. Yes, nobody's able to recall it. Yes, everyone. And you also asked me certain doubts related to it and you wanted to cover it. What was that in this week's lecture? We majorly studied about sample preparation last week, and there was one lab session too, which you all wanted me to cover as well, right? Yeah. Okay. So I think we can start with the lecture. It's already 8 minutes up. So I was just waiting if someone wants to join in. Mm, Okay. Yeah. So welcome to week three of NPTEL course, which is proteins and gel-based proteomics. This specific course has a code of BT57. So today's lecture is about week three lecture. So as just to tell the introduction, so in NPTEL we have every week there are 5 lectures which are generally uploaded. You all go through that, and if you have any doubts, we as TA try and solve those doubts for you all. In week three, Sir majorly talked about two-dimensional gel electrophoresis technique, how exactly that is performed, how samples are prepared, how it is used, applications, etc. So in this week three, we'll be dealing with the how exactly this specific technique is performed, what all steps are involved in it, in the first dimension, then in the second dimension. So we already started with this. If you all remember, we just did the first dimension of the two-dimensional electrophoresis. Yes, am I right or no? So we'll be dealing with that. We'll be talking about second dimension as well, and then we'll be talking about how those after the run, how those can be analyzed, a bit of applications and challenges of it. Before going to that, in the last lecture when we were dealing with sample preparations, during that you people had doubts regarding the lab session which was taken by one of the speaker from DNET, I think so. So what he was trying to tell, like along with that, there was one more lab session. I think that was easier to understand how exactly uh, this two-dimensional electrophoresis is performed. I don't think you people had doubt in the second lab session, but you majorly had doubt in the first lab session, right? Am I right, people? The second session was just, I think, 15 minutes or so, and in that, people like the one of the lab member has shown you all how exactly isoelectric focusing is performed. Yes, have you all gone through those lectures? Yeah, so you majorly had doubt in terms of the first lecture, first lab session lecture, right? Yeah. So what I did is I tried to make it in a same like make it a little bit simpler version and made a PPT out of it. So this this deals with tissue preservation technology and a bit of sample preparation. There was one of the presenter from DNet or AB. So basically, that's a company which deals with making stabilizers instrument. So that is required in terms of sample preparation because of various reasons which we'll be dealing here in this particular first session. So what So majorly what he was trying to tell you is this specific sentence like the results are never going to be better than your sample. So depending upon your sample, so you as so as good as your sample, your results will be as good as that your sample itself. So your sample should be the most accurate one. And for making a good sample, you need to take care of certain things. That's what this specific professor tried to tell you all. So if the sample is of low quality, so regardless how much you do the analysis, how much money you spend on doing a mass spec or doing any kind of technique, that will still going to be a low quality because the sample itself, because the main source by which you're getting the data, that itself is not that great. So in order to make sure that your sample is the most stable and has been acquired in a more stable format, and it is whatever components are present in the sample are stable enough or detectable enough so that you get the good results also, like by spending this much amount of money in doing mass spec. So generally, uh, mass specs are expensive, but they are only as good as the input sample. So if your sample is good, your mass spec data will also be good. But a lot of people generally focus on like if I'm doing mass spec, then my results would be good automatically. Although mass spec will try and get each and every single thing out of your sample, but it will only take what your sample is telling about. Like for example, you have acquired the sample, but the way you are acquiring it was not that good enough, and there was some degradation inside the sample, etc. So mass spec will give you those results itself, like whatever is present in the sample, it will give you. So if your sample preparation is good enough, your mass spec will also give much more accurate results. So what that professor was trying to tell you is like what happens after sample collection. As soon as you take out the sample, what exactly is happening inside those cells? Like when it is outside the human body or outside the any organism, whatever your sample is. So here we are majorly dealing with, for example, blood sample. So inside the body, the way blood is going through, and outside the body, the way it is going through, it's a little different. So cells in the sample are still very much alive, and they don't know that they have been removed basically. So there is loss of blood flow, so blood transport, so oxygen and nutrients are not getting exchanged as such. That immediately stops as soon as you take out the sample. There is hypoxia, there is no oxygen, there is ATP depletion as well because glycolysis, phosphorylation, and all those is not taking place. So energy is not being there. So all these changes happens as soon as the sample is out of the organism, basically. Because of this, there is lactic acid accumulation, and because of that, the pH drops. And if pH is getting dropped, a lot of enzymes will be affected, right? Because or basically a lot of proteins will be affected because the structure of a protein or the way protein can function highly depends upon pH. So if that is going low, it will affect your sample, like the proteins present in the sample. And there will be cellular stress because there is low ATP, there is low pH, and because of that, whatever it is alive of, it will try and send those signals that all this is happening. So try and make sure that cell responds by changing phosphorylation, digesting proteins, and altering lipids. That's what cells tries to do. So that to survive, because it still want to survive, right? In order to survive, it keeps on changing this stuff. So what you need to do? So as the post sampling changes occur in two distinct phases. One is sample collecting and free stabilizing, or during homogenization and extraction. So as soon as you collect sample and you freeze it, even that will have some effect on the sample. Or else, as soon as you are collecting the sample, you will try and lyse the cell, extract the proteins out of it. Even that will have some effect. So basically, from the moment of excision until preservation, cells actively respond to stress, whatever it is undergoing through, like the temperature it is going through, the way you are handling the sample, yeah, either you are shaking it, vortexing it, centrifuging it, whatever it might be. So during that process, phosphorylation change, proteins will degrade, and every minute is responsible for certain change in your sample. Similarly, for example, while you're doing the extraction, so you'll add certain inhibitors, you'll add urea, you'll add organic solvent to lyse the cell, etc. So all this will be responsible for additional changes to phosphorylation, peptides, lipids, etc. So although you are thinking that your sample is safe because you are adding this, but due to addition of this, there are a lot of changes being happening inside the sample. So there are three classes of molecules that change rapidly, that is peptides, protein phosphorylation, and free fatty acids. So we are not going in detail of that, but these are the stuffs which generally keeps changing. So all these things that professor tried to talk about in each and every section of the uh, type of protein or a type of molecule which can get degraded as soon as you or like basically a lot of changes will happen because it can increase or decrease depending upon the tissue type, depending upon the type of protein it is, the phosphorylation can increase or can decrease, and accordingly in your sample mass spec will detect in that way. Similarly for peptides, so in terms of neuropeptides, they rapidly degrade as soon as you take the sample. So the amount of neuropeptides, for example, your study is based on neuropeptides. So studying this will be much more difficult because they generally get degraded as soon as you take the sample. And imaging will also confirm the degradation fragments only because of the non-stabilized tissue. So MALDI is basically a detector, the ionization source of the protein sample, and that helps you all for detection of samples. So what can be done in order to avoid such issues? So this specific company, Denator AB, has some heat stabilization technology, and what they do is they try and heat the sample as soon as you take the sample out. So that's what helps them to be in the state at which everything can be detected. So you heat the sample at 90 degree, which causes irreversible protein denaturation. So it will denature the protein. So although in your external processes you will ultimately degrading the protein, right? Like denaturing the protein, not degrading. So accordingly, the samples take 20 to 25 seconds for small biopsy, and you need to heat it first. That's the first condition, temperature. And depending upon the type, is it your solid tissue or a liquid unit, this specific system is present depending upon. So it is in terms of solid, that is dome shaped. So this will depend upon your sample, that type of instrument you need to take it. Or if you are dealing with dried blood spot, so this depends upon the type of sample you are having. You need to have that cartridge system so that it can handle your sample. Just a minute. Yeah. So it can also be used for frozen samples as well, and it can stabilize the fresh or frozen samples. And so there is some exception with this. That in terms of RNAs, RNAs are very short and held by disulfide bonds. They refold after cooling. So nearly all other enzymes find a stable denatured state and do not recover, but RNAs won't be able to do that. So except that, all other things would be much more stable comparatively if you heat it as soon as you take out the sample. Like basically, this will cause irreversible protein denaturation, and then after that, all the proteins are there. So basically, they are not getting degraded or getting phosphorylated or getting like there's no changes being happening inside them because you are heating it right away. Similarly, there is clinical integration as well can be placed and downstream compatibility like Western blot assays, whatever you are doing later. So it will be compatible with that. So what they have found is in terms of phosphatase activity, the heat stabilized didn't give much of a background, and all those so phosphatase activity was checked. Similarly, kinase activity was checked. So in terms of heat stabilized sample, 99.6% of kinase activity was abolished. What does kinase do? It adds phosphate group to the to your protein, right? So as soon as you heat stabilize it, so kinase activity was abolished completely. So it will not add phosphate to any of it, indicating that whatever proteins are being there, they will not be changing. So that's what heat stabilization will do, and it will try and help that the actual sample you are collecting, the proteins are present in exact state of the same state. So they also done phosphoproteomics and tried to check heat stabilized versus snap frozen. So generally people snap freeze it and hope that it will give you the same amount of proteins which are present in the sample. But what they found that when you heat stabilize it immediately, they found much more higher amount of phosphopeptides compared to the snap frozen ones. So generally people follow this. Like they take out the sample, add all of these and snap freeze it, and the number of phosphopeptides found were much lesser compared to heat stabilized ones. So if you do it as soon as it uh, possible and heat stabilize it, you will end up getting more number of accurate proteins compared to that of snap freeze one. So this is just clinical integration. By doing that sample, you can do make biobanks out of it. The downstream applications, all that can be done. Like the basic aim is that as soon as you take out the sample, heat stabilize it so that more amount of accurate sample analysis you can be doing. Because as initially in the first part of the lecture we are talking that it keeps on changing. As soon as you take out the sample, because of the cellular stress, the proteins inside the cell keeps on changing. So you need to stabilize it. That was the overall aim of what sir was trying to tell you in terms of this specific lab session. I hope it's clear now. Like if you have gone through those lecture, you might be able to relate it. Yes, are you all getting it? Yeah. How about others? So that's what. So these are just key takeaways of the same thing. Overall aim is just basically your sample quality should be as good so that you get better MS analysis. Post sample changes begins immediately, and there are two danger zones of it. So you need to make sure that that doesn't happen because of its stabilization, and most of it gets reduced because of doing this specific step. And there is one more reference you can search freely online. There's 11 golden rules for proteomics. So that's what it was about in terms of lab session one. Okay. So I think now we can start with the week 3 lectures. I'll be going as fast as possible because we took a bit of time in terms of lab session thing. Okay. So in week 3, as we already did a bit of uh, understanding about how samples are prepared, like after the sample preparations are done, one of the way of analyzing the sample is two-dimensional electrophoresis. So this is the basic workflow of two-dimensional electrophoresis, wherein the first dimension is isoelectric focusing. Then you equilibrate the same this strip where you have done isoelectric focusing. Then the third step is doing the second dimension, which is SDS page. Then after you run that, we already done this one dimension thing of SDS page, but when you're doing 2DE, you can directly just a minute. Okay. So we did one dimension wherein of SGS page wherein we do stacking gel and dissolving gel, but in terms of two-dimensional gel electrophoresis, you can there's no need of stacking gel because stacking is done with isoelectric focusing, and you can directly do the resolving part in SGS page in second dimension while doing this technique. Then how there is different ways of staining depending upon the sensitivity and the concentration of protein you are having. You can accordingly change the staining method. Then visualizing them will accordingly change. Then how do we analyze those samples? Then spot picking can be done. Like if any specific protein is highly upregulated, you can just take that to enzymatic digestion and do MS analysis. We'll be trying to understand all of this in this specific lecture. How can we do it? So, so basically study global and differential protein expression can be done with the help of this specific technique. You can dissolve proteins from complex mixtures, that's obvious. And it will also help you to study isoforms and post-translation modification, depending upon your staining technique, basically. And visual analysis of proteins can be done. So this is the overall picture of two-dimensional electrophoresis. You have a pH gradient strip and you go with first dimension, that is isoelectric focusing. You run the sample in this. So depending upon the isoelectric point, the protein will stop moving. So its protein moves till according to pi, and as soon as it gets the pi, which is isoelectric point, the point at which protein has net zero charge, at that point it will stop. And that's how proteins will be separated in isoelectric focusing. Then you need to take this same thing, align it in a uh, horizontal format in order to run the gel vertically. And as soon as you do keep it, you can do SGS page, and the number of proteins which are of that specific PI will get separated, and you will get a more better separated proteins in terms of doing 2D gel electrophoresis as compared to one-dimensional SGS page. So I just wanted to ask. So okay, I'll ask that question later after completion of one this 2D gel electrophoresis technique. So that was just an overview or gist. We'll go in detail about how exactly this is performed. There are few steps after this in order to go ahead with SGS page. We'll talk about that and then we'll talk about SGS page too. So isoelectric focusing, it's a protein suppression technique. It depends upon solely depends upon pi, which is isoelectric point. It's the point at which it has zero charge, lack of charge basically, and it does not move in electric field. And this is how you can identify pi of each and every amino acid. And for a protein, each amino acid's pi will average out for for uh, for you to understand what might be the pi of that whole protein. So just to give an overview, so for example, initially if a protein is positively charged, until and unless it gets reaches a point at of pi where it has plus and minus, which will cancel out and it will be zero charge, it will stop moving. Similarly, the blue one here stopped moving here, and the red one got stopped moving here. Similarly, you can see the yellow one here. It was here initially, and it went on to move ahead until it achieves net zero charge. That's how it keeps on moving until it achieves zero charge. So proteins, initially you need to do the rehydration step of IPG strips. So proteins extracted from a previous step, which is sample preparation, which we learned about. So you need to rehydrate. So as soon as you have IPG strip with you, IPG strip is nothing but a strip which is having pH gradient. This specific strip is called as IPG strip. So that you need to rehydrate overnight in a reswelling train at room temperature, and that rehydration will have specific IPG buffer. And passive rehydration, no voltage applied, and active rehydration. There are two ways of doing rehydration, passive and active, wherein voltage is applied or not applied. Overlay mineral oil on rehydrated strips in order to keep it like contamination free. So and also to prevent evaporation. So IPG strips are supplied with dry and stored temperature minus 20 degree initially. and samples should be loaded on strip after thawing. So it is generally kept in -20 those strips in order to maintain the pH of to through the gradient. And there are different strips are also available depending upon there are 4 to 8 or 3 to 10. Depending upon your requirement, you can have such kind of pH strips. And samples can be loaded either with cup loading or direct loading. There are two different methods for loading. You might have seen all of these types of method in this lab session too, where people try to explain how isolatory focusing can be done. So as just mentioned, there are different ways of different pH strips are available of IPG strips. Similarly, they are of different lengths also, and accordingly, such kind of SGS page needs to be used depending upon the IPG strips used for separation of in the first dimension of this specific method. And so large gels are recommended to resolve spots better, or else. But there is one problem with large gels that it is difficult to handle and it is tedious also. So you need to make sure how much you can handle, how much your protein is, whether you need much more resolved bands, because if there are few proteins which are very much close in terms of their molecular weight, then you need to have some larger band so that it gets resolved properly and you can spot that specific protein out of it. So depending upon your need, you need to choose that. Similarly, uh, the protein loading volume will change depending upon the strip you are having. For example, for 7 centimeter, the dyeing which can be done, it can be of 160 microliter approximately. If your strip is much more bigger in size, then you would require 350 microliter of it. So this is separation in isolatory focus is on the basis of isolatory point. Just to uh, revise the same thing. Sample loader strips overnight, then you need to rehydrate. After overnight rehydration, the first step is rehydration, and then you load the sample, you run it, then protein mixtures will migrate depending upon the PI, and it will stop as soon as it reaches PI. So after doing isolatory focusing, your that's after first dimension. You need to equilibrate the membrane, basically IPG strip, by coating it with SDS page by cleaving inter and intrachain disulfide bonds. That's what SDS will do. And sorry, that is not one which SDS does. You need to do reduction and alkylation. That's what reduction will do, the breaking of disulfide bond. And you need to alkylate it as well. So those two steps are done in terms of equilibration. So what happens in this equilibration? You add this solution which has SDS, DTT, etc. And this basically, you need to saturate this IPC strip with SDS and reducing agents. These are reducing agents, urea and DTT. SDS will give a charge to the negative charge to the protein, and DTT will try and reduce it, or urea and DTT. And alkylating agent like iodoacetamide will alkylate the reduced proteins. And what is happening in this specific step is equilibration. You need to do twice 10 minutes each, the first equilibration and second equilibration. So what is happening during this equilibration is, so if this is your specific protein, you add the DT2 to it, which will break this specific, which is dilthiazolidine, which will break this disulfide bonds. Now it is separated. Now, in order to avoid this renaturation of or rebuilding of this disulfide bonds again, you need to alkylate it by adding iodoacetamide. So that is alkylation taking place with the sulfide group here, and this will help so that it will not rejoin again, or it will not make the disulfide bonds again. So you need to equilibrate your proteins. So what is happening during equilibration? Those proteins are, whatever disulfide bonds are present, they will break down, and it will also have been alkylated so that they are not renaturing it or making the disulfide bonds again. That's what is happening in equilibration step. So that is the step you need to do after your first dimension. Then in terms of second dimension, that is SDS page. So before talking about this, they also showed a bit of practical uh, thing how exactly gels are made and how they are equilibrated, etc. So this is about gel casting thing. So there are few. This is how IPG strip looks like. And this is a tray wherein you equilibrate it, so you can keep the IPG strip in this tray and add the equilibration solution for 10 minutes each. You need to do it twice. So that's how it looks like. If you have gone through the lectures, it would be easier for you all to understand. This is the IPG strip. You are loading your sample here, and that's how separation happens. Then the same thing, uh, you can add it into buffer tank. This is how huge the buffer tank looks like. What happens when you add SGS? It is giving negative charge to the protein. And when you are running the sample on SGS page in the second dimension, it runs on the basis of molecular weight, etc. So more majorly this lecture was practical, like people have shown how exactly there are few lab members who have shown you all how exactly this technique is performed. So I can go through those specific practicality of this specific first dimension and second dimension separation. So in the next lectures, I talked about staining and destaining method. So you have run your first dimension, you have run the second dimension. We have already studied in detail about SGS page, so I'm not talking details about SGS page here. So after running the sample, you have done your first dimension, you have equilibrated it, you have run the second dimension. Then you can stain it using by different ways. One of the ways by adding comassive blue. So it is biosafe comassive stain and it is ready to use. You directly get the reagent now available. And this used stain can be disposed as non-hazardous waste as well. Now you have this completed gel which has run on the second dimension. You can just add it to the staining solution. Is anyone asking me anything? Okay. So so this is commercially available. You can just use it, and this is how the Comasi blue stained gel looks like. Similarly, there is silver stain, which is more sensitive than Comasi blue dye. For example, if you are having any proteins, and if you want to find any protein which is less concentrated, like maybe present in nanograms, then you can use silver staining instead of using Comasi blue. So this silver staining, basically what it does is it has silver amine complex bound to tungsto silicic acid, and this specific ions transfer this acid to proteins. And formal there is in the like in this process itself, you also add alkaline solution, which is formal, which because this alkalinity is because of formaldehyde present, which reduces silver ions into metallic silver, and that's what helps you all for detection while visualization. And this whole reaction is stopped with acetic acid. So that's the whole protocol while doing this silver staining. In terms of combative blue staining, you just need to add this combative blue stain it, and you need to de-stain it as well. So because this staining happens, and it will stain everything present in the gel. It will intensely stain the proteins present in it, but it will also stain the gel, the whole gel. While doing de-staining of the solution, in that process, whatever the gel is being stained here, that will get destained, whereas protein will not get destained because it forms complex with the protein. Whereas in terms of silver staining, this is the whole protocol you need to do. Like you need to add alkaline solution to it. Then in order to stop that reaction, you need to add acetic acid. So all this specific protocol is done uh, while doing silver staining. It's just the reaction which is taking place when you add silver stains. Then there is another type of staining like cypro-ruby, wherein fluorescent, which is a fluorescent stain technique, and it is visualized under UV. So the benefit of this technique is it gives little background as compared to these other two techniques, comassive and silver, and it is also able to detect glycoproteins. So as we were talking about post-translation post-translation or modified proteins, if your study deals with that, then you need to use such kind of stains because silver and comassive blue stains will not be able to tell you any glyco-related protein or phosphorylated protein, etc. And it also helps you all to detect low molecular weight proteins, such kind of dyes. And it is also compatible with mass spec. So this is basically methanol acetic acid in order to stop that reaction. And when you are visualizing using this dye, you need to have maximum excitation wavelength and emission wavelength at this. So you need to set a protocol accordingly in order to visualize a gel which is stained with cypro ruby, and you can visualize the gel. There is another dye which is ProQ diamond, wherein it is also a fluorescent dye and it helps to detect phosphorylation. For if you have your study deals with phosphorylated proteins, then you can use this specific dye in order to detect phosphorylated proteins. So in that specific procedure, there is a washing step which involves. So this is destaining. This washing step is nothing but destaining though, and this is how the gel looks like in terms of ProQ diamond. So you can also do dual staining technique as well, wherein diamond or other post-transition modification detection stains can be combined, like cyproruby, which detects glycoproteins. Diamond detects phosphorylated proteins. So dual staining allows more than one detection protocol on a single gel. You can also do such kind of staining as well. So as I was talking about, like silver stain can even detect nanogram level proteins, and all are MS compatible, whereas Comace Blue deals with a protein which can detect around 40 or 10 nanogram. Whereas if you have much more lesser concentrated protein, you can use silver chain dyes. So we in this lecture, sir majorly talked about SGS page, gel casting techniques, etc. If about different dyes and comparison between them. And he also sir also gives you references if you want to deal with more details regarding that. Then how do we analyze this specific uh, gels? Like once you have the gel with you all, how do we analyze that? So for analyzing, there are certain instruments available like densitometry. So densitometry can tell you about, for example, you have run the sample. There is one specific protein which is highly dense in that specific lane compared to other proteins, or else you can also detect in all the wells itself, there are few proteins which are very much dense. So you can tell about how much percent it is more compared to other proteins. Whether if you are dealing with one specific lane itself, if you have added the lysate itself, for example, you have lysed the cell and you have added the cell lysate, and one of the specific protein is highly expressed compared to other proteins. So what you can do tell is, for example, 40% of uh, the lysate is related to that specific protein, or 50% of it is related to that protein. You can at least compare it with the whole thing and tell that specific protein is much more concentrated compared to other proteins. That's what densitometry will help you all to tell you, and it will also help you visualize. Similarly, there is imager, typhoon variable mode imager, which will also help in visualizing the proteins. So, sir talked about the software as well, how exactly your proteins can be analyzed using this specific software. Just a minute, people. Okay. So, sir, talk a bit about software which is used for understanding the sample or analyzing your results. So in terms of 2D gels are scanned using those scanners like we just talked about. For example, Typhoon, which one of the imager which helps you all to compare two gels and detect what might be the difference, etc. So this software, what does it do? It helps in spot identification. It helps in comparison of gels. It helps in overlaying both images also. Like overlaying will directly tell you like what is the difference between two of them, right? It also helps you cropping the gels. It helps you all to do statistical analysis, etc. So, for example, the crop tool. So it will. So whatever your major protein of interest of what specific molecular weight and what might be the PI of it. So there was P. Initially, you have run the samples depending upon PI, right? And then you have run the sample depending upon molecular weight. So you know that at what PI, at what molecular weight your protein of interest might be, then you can accordingly check or analyze that specific block itself instead of analyzing the whole gel, which will have a lot of proteins in it. So you can just minimize your analysis of your interest itself. That's what crop tool will help you all to do. So it allows specific defined region of the gel to be cut and then analyzed. There's a zoom tool as well, which expands a specific area. And if there are two proteins very close to each other, then zoom tool will help you all to check whether if they are separated or they are together, etc. And image overlaying, that's obviously will help you all to compare two spots, whether they are similar in size, larger in size, whether they are at the same molecular weight or different molecular weight, etc. So separate images are overlaid to appear as single merged one, and both will have different colors, so you'll accordingly will able to analyze whether it's at exact same location or different. Then spot analysis. So these peaks are nothing but the intensity of which I was talking about, which is a property of densitometry, and I'll tell you which what might be the uh, concentration of that specific protein in your sample. So it is possible to obtain physical and statistical parameters for each spot on gels and allows the gel comparisons spot by spot. You can do the comparison like this spot comparing with this spot comparing with this spot. You can do that. So, so blue and yellow mixes together to become green here. So this are present in both the sample, whether this is uniquely present in this sample only, whether this is uniquely present in this sample only. So that's what overlaying will help you all to understand. So just to give one example, this is healthy control and this is disease control. So you can compare like these specific proteins here. In healthy, it is very much less compared to that of this in the disease sample. Similarly, in healthy, these are much more highly upregulated compared to that of this in the disease sample, which are lesser in concentration compared to this. Similarly, there are few proteins present here which is not present in healthy control but only present in disease sample. So you all can study these specific proteins by using mass spec and try and check whether they were the one who are actually responsible for the disease, etc. Something like that. So there are different softwares available. These are few softwares which are available, and sir has also provided websites of it if you all want to go through that anytime. So one of the step is gel matching when overlaying will help you all to do that. So how much it is upregulated or how much it is downregulated. So plus and minus indicate that. So compared to this, this is much more greater plus 2 this is +1 compared to this. So accordingly, you can do the analysis of it using that software. How many spots can be seen? How much is the intensity? How much is the area of that specific spot? All of this can be analyzed using this specific software, and it gives ID to each and every spot too. Then you can create different graphs out of it and check each and every spot separately at the same location, whether that specific protein was present or not present in gel 1. So you can detect the PI at which it was stopped, and how it ran over the course in second dimension. You can detect all of this. What could be the molecular weight of it? What is the area of that spot? What is the volume, etc.? How much is the intensity? All of this can be analyzed using that specific software. So it helps you all to do global protein expression analysis. So if you're doing overall analysis of the whole gel, that's global protein analysis. It will help you to differentiate protein expression by comparing the two gels. You can differentiate between them. You can resolve proteins from complex mixtures, that's obvious. And it helps you all to study isoforms as well, post-transition modification, depending upon your staining technique. It will help you visually analyze the protein. So overall, protein profiling can be done using this technique. For example, condition A and condition B. There are two different proteins. You are running the gel, solubilize the proteins from cells, you will solubilize it, prepare your sample accurately. You will first run the sample on isoelectric focusing. Then, according to the PI, depending upon your first condition, it will accordingly get separated. You will run two gels of one condition A and condition B. Similarly, you will do condition A and condition B. Depending upon your need, you will do the staining of those SDS gels. Then this is condition A, this is condition B. So while doing analysis, you can exactly spot like this specific condition has led to some differential protein expression compared to that of this condition. So that's what, like, this is one of the example how you can application in terms of how you can use this specific technique to understand that. This is the same thing, like you can analyze the intensity of it, do statistical analysis, etc. So there are few more applications which are talked about in his fourth and fifth lecture. For example, host response to malarial infection. So, so in this specific example, they have done serum proteomic analysis where they have taken samples from a healthy control and a patient who was suffering from malaria. They have taken the samples, separated the plasma and serum, and serum proteomic analysis was performed. So there is healthy control, there is malaria, the person suffering from malaria. Similar to that of condition A, condition B. Here, healthy control by the malarial person. But here they have done MS analysis as well. But we will be talking about gel-based proteomics, what they found in terms of that. So generally in serum, these two are, we did talk about that during sample preparation stuff. So these two are highly concentrated, albumin and immunoglobulin. So you need to separate that by using affinity column or ultrafiltration, etc. So will be removed, and then can do the protein analysis of this low concentrated samples. So serum sample, it can be crude serum, desalted, then sonication and desalting, then abundant protein removal by desalting. You need to do that. Then 40 microliter of this is a protein extraction protocol. That serum is taken after removing the uh, highly concentrated stuff like albumin and immunoglobulin. Then you can incubate. So this is just the protocol of it, like how exactly proteins are extracted out of serum, which we also done during the sample preparation steps, like lecture. So depending upon how you are taking the sample, the number of spots which were obtained by effect of different sample processing. So this is again deals with the sample preparation stuff. So taking directly the serum itself gave you a lesser number of proteins spots basically by doing IPG like these two dimensions. Whereas if you have desalted it properly, sonicated it properly, depleted the abundant proteins, etc., the number of spots were much greater. So that's what even this talks about the week two thing. It's week two lecture itself. Like sample preparation is much more important in order to get or detect all the proteins present in the sample. So protein extraction, depending upon acetone versus TC acetone precipitation. So that gave higher number of spots, TC acetone compared to that of just acetone. So you can compare methods and check which method is most suitable. Similarly, crude serum or depleted serum. So crude serum will have albumin in higher concentration immunoglobulin, but once you separate that, so depleted gave you much more separated proteins and because they are interfering with the separation, right? Because they are present in high concentration, whichever are present in lower concentration, they are interfering with that, like during the isolatory focusing or during SGS phase. That's the reason they were not able to get separated here, whereas depleted serum, those proteins were clearly separated. Similarly, chromatography staining and silver staining. So silver staining will also help you to detect. So these proteins are more low, much lesser concentrated. You can see few spots here as well, few spots here too, etc. So silver staining helps you all to determine much more lesser concentrated samples as well, like 1 nanogram and stuff, whereas this deals with 40 nanogram. So it detected 995 spots compared to 919 spots. So around 70 proteins were detected more when you use silver staining. So this is the same comparison method comparison, crude, sonicated, the one with the TC acetone, and we also studied about trisol method, if you all remember. So accordingly, the amount of protein loaded, although there was a change, but the number of peptide spots identified accordingly differs depending upon your sample method you are using. So this is they took 8 samples of uh, Vivax malaria, 8 samples of healthy controls, and then they compared it in 8 lanes, and this is how it looked like. And so if you see, there are a few proteins they wanted to identify whether those are the one which are responsible for the Vivax malaria. Like basically, the condition has caused certain proteins to be overexpressed or downregulated. So when they tried to study that, differential spot expression were identified in like these initial 3 samples compared to that of healthy control, whereas those specific proteins were not present, whereas in Plasmodium vivax individuals, those specific proteins were present. Now, these specific proteins can be cut from the gel, digested, and subjected to mass spectrometry to detect what protein this is. All those can be done later. So, this there were few proteins which were found down-regulated in the uh, diseased individual compared to the healthy control, whereas few of the proteins were upregulated in the diseased one compared to the healthy control. So that's what all those studies can be done using this technique. So basically, here we are talking about applications of 2D gel electrophoresis. So you need to have all of those 2 samples. You can compare them and check which proteins are upregulated, which are downregulated. Then you can take that specific spot, do mass spec analysis, and try and find what is the exact protein. So here you just know the molecular weight of the protein and isolatory point of the protein, but you don't know exactly what protein it is which is causing this or which has upregulated or downregulated. That can be done using mass spectrometry. So using mass spec, having standards, you can exactly determine what type of protein it is. So in this one of the specific study, they found that these were the proteins which were down-regulated, these were the proteins which are upregulated. References. Then Sir also discussed two case studies in his next lecture, effect of drug treatment on malarial parasite as well as SARS syndrome, plasma proteomic analysis of SARS patients, as well as the challenges associated with two-dimensional gel electrophoresis in his last lecture. So this is one of another application of it. So in one of the conditions, this coatom is a combination of artemisinin. So this is a specific drug which is being given. So we are studying effect of drug treatment of malarial parasite. So this is the specific drug which is being given, which is a combination of these two drugs. And so drug of choice for all cases of non-severe malarial worldwide, which is this one, coatem, and it is mediated specifically through endoperoxide moiety. That's how the mechanism of it is. And this is the general life cycle of the plasmodium. Then from a person, the uh, proteome, when a person suffering from this specific disease of Plasmodium falciparum, they try to analyze after fractionation. So fractionation is a step in sample preparation itself, if you all can recall it. So the time point at which these are being collected, accordingly it will change. So you know the life cycle of uh, Plasmodium. So accordingly, it is necessary that it is being collected at that time point. So synchronized parasites isolates from host erythrocytes. So it is, if you all know that RBC is the where location where these specific parasites inhibit, and that's how malaria is caused. And this is basically a protocol which you need to follow while collecting the sample. Then they are loaded, separated, etc., which we were talking about, two-dimensional electrophoresis. So this is the mechanism. So when these two drugs are given together, so glycolytic enzymes, so reaction with late ring stage of Plasmodium falciparum. So it reacts with this specific uh, stage, and glycolytic enzymes are released out of it and inhibit the propagation of that specific Plasmodium. So when they tried to do this, so they found this few of these proteins were upregulated with this specific drug, whereas with this specific drug, all of them were upregulated, whereas in this using this drug, these were downregulated. So depending, so there is an effect of drug on proteins present in that individual. So that's what they tried to study, and they found that few of them were upregulated and downregulated depending upon the type of drug being given. So stress response proteins both were upregulated in both of these conditions, whereas glycolytic enzymes were downregulated with this drug and upregulated with this drug. So the major finding was alteration of parasite proteomes induced by two components, these two specific drugs, and established relationship between pharmacological relevant concentration and time of exposure for the two components. So accordingly, with the help of this, people can now design a specific strategy in order to make sure that it gets completely downregulated and both of those plasmodium dies. So that's what they try to find alteration of parasite proteome induced by two components, an effect of two anti-malarial drugs in pharmacological relevant conditions. That's what they studied, and membrane-bound calcium binding proteins were seen upregulated due to both of these treatment. So basically, this type of studies will help you all to design a specific treatment strategy. Similarly, there was one more application we talked about SARS plasma protein analysis. So generally, this is how SARS virus looks like. It has glycoproteins, spike proteins, it has nucleic acid phosphoprotein, and it has RNA for its replication, and it causes. That's how it spreads. You all know about SARS obviously because of COVID. So, four SARS patients were taken, 22 plasma samples were collected, and six healthy patients with six healthy controls. So, we tried to compare with the diseased one with the healthy control, and they tried to find which proteins were upregulated or downregulated, and found which were the one which might be responsible for this disease. They found that in healthy control, was one specific protein which was highly upregulated, whereas in disease it was downregulated. Whereas these two proteins were downregulated in healthy control but were upregulated in the SARS condition. Then, so basically, how mass spec can be used? You can just cut this specific band of protein of interest, take this protein, do in-gel digestion by using trypsin or any protein digesting uh, enzyme, then it will make certain peptides out of it. So this protein will have unique peptides which will be solely able to detect what type of protein this might be, and that can be done with the help of mass spec. So there are different mass spec based studies. So here we can see quadrupole mass spec, wherein there are four poles which helps in uh, fragmentation of the protein coming in contact with the ion source. So this is mass analyzer 1, this is mass analyzer 2. So this is MS1 and MS2, indicating, for example, if there is one specific protein of, for example, 180 kilodalton, and that will break into two components in MS1, and those two components specifically, for example, 180 kilodalton has been separated into 180, for example, 100 and 80. So it will in the collision cell we can set up a specific term that it needs to select only 100, or it can select only 80 or whatever. All those protocols can be set up, and as soon as it goes into the second analyzer, that specific 100 will be again fragmented accordingly, maybe 60 and 40, for example. Then accordingly it will create a specific mass spectrometry like this, and you can see those specific fragments and try and analyze the protein of interest by matching it with the library. So it will change depending upon the reducing and non-reducing conditions. So this we also talked about during question and answer session in the last lecture when we were talking about one-dimensional SGS page. So because of this in reducing agent, this got reduced into 2 here. So, so acute phase proteins in excess, whereas indicates bad liver responsible for weak defense against SARS. So all this can be analyzed accordingly. So what are so these were the few applications, like Sir gave 3 to 4 applications which can be used uh, by like doing this specific technique, 2D gel electrophoresis, and people can try and understand what proteins might be responsible, etc. So, what are the challenges associated with 2D gel electrophoresis? A good 2D gel looks something like this. All of them are separated, and each spot can be visualized separately, and you can accordingly cut the specific protein of interest as well, and do further analysis, etc. So this is how a good gel looks like. Similarly, this is also a good gel. So major sources of variation, like if everything goes well, this is how it looks like. But if something is wrong, so gel artifacts or gel to gel variation can happen because of during your image analysis defining spot boundaries. Like you need to define spot boundaries while doing the analysis. So that manual errors can take place. User bias, like one of the student is more prefer to one of the protein, whereas another preferring one something else. User bias can happen. All this can be some major source of variation in 2D gel electrophoresis. Sometimes you might see streaking like this, and if you are seeing something like this, it's majorly because of trace impurities. Your buffer has trace base, and if that specific trace base is not pure enough, you might see protein bands like this. Similarly, while making SDS page, if you haven't used IMED properly, the polymerization will not take place accurately, and because of that, the proteins will get separated, something like this. And so all this, for example, you are using crude sample itself, and if you are not fractionating it properly, your sample preparation is not that accurate. You might see gel something like this. So all those are some limitations or challenges related. So that you need to make sure that your protein is much more accurately prepared. The type of staining you are using, type of the way you are making your gels, etc. All those are good enough so that your results are good. If you are doing solubilization without precipitation, your gel might look something like this. So if you have only equilibrated with DTT, the disulfide bonds, if you have not alkylated it, the proteins might get uh, again it might get annealed again with the sulfide bonds, and it might give you some separation, something like this. If you have over focused your gel while visualization, it might look something like this. So it also depends upon your source of sample between individuals, between biological states. So everything affects which can't be controlled. So all this cannot be controlled though, but the way you are analyzing your sample, loading your sample, making your samples, preparing your samples, or whatever method you are using, isolator focusing or 2D electrophoresis, what are components required for making them, the way you are making it, everything will have some effect on your variation. So variation in sample preparation, IP-specific pH range, presence of salt, it can change the way your gel looks like. The voltage applied while separation, if you are running your samples much more faster, like in 200 volts or something like that, it will be different as compared to that of if you're running your sample at 100 volts. Equilibration time, quality of chemicals used, everything will have some effect on the way your gel looks like. So these were the few challenges associated with 2D gel electrophoresis. So those were the 5 lectures which sir talked, like I just gave an overview of it. What the fuck? I'm sorry. I think I got logged out somehow because my net got. Hello everyone. I don't know when did I logged, like it got logged out automatically. How much did you all listen to? Because I was speaking continuously. I didn't see that it got logged out. How much did you all see? Till what which slide? Can you all see the screen though? No. Yes, people. I didn't know that it got logged out. I was continuously talking. Can you all see the screen now? And kindly let me know till what time were you able to see the lecture. Although it is getting recorded, so yes, please let me know. Till what slide you were, like we were online? Did you all listen about SGS training? Did we all listen about different applications of two-dimensional electrophoresis? Malarial patient, healthy sample, COVID sample plus healthy patient, etc. Yes, yes, everyone. Healthy versus affected. So I'll just I'll stop and present again just a minute so that I have this window so that I can see what you guys are talking about. Yeah. Okay. So I just completed though, but till what slide were you able to see it? Did you all were you able to see this as well, the COVID thing? I mean the SARS thing. Yes. So these were basically different applications of did you all did we were you all there when I discussed the effect of drugs on treatment? Yes, yes, no, yes, no. Yes or no? People please inform so that I can accordingly till what slide you were there. Like effect of drugs were done or you all were there till here, like the few were up regulated, few were down regulated. Sorry. So were you all there? Did you all when I discussed this effect of drug treatment on malaria? No? Okay, before that you all were there, right? During when I was trying to explain these spots. Yes? Okay. So so last lecture is left basically. Okay. So in this, sir gave few more applications related to it, related to the same two-dimensional electrophoresis. You gave few case studies, where in one of the study, so basically this specific drug is being given for treatment of Plasmodium falciparum malaria. So it's a combination of two drugs. artemether, and lumefantrine. So this drug of choice is for all cases of non-severe malaria worldwide. This is what is given, and so it depends upon the life cycle of Plasmodium falciparum, the time point at which the specific drug is being administered, and so synchronized parasites isolated. So RBC samples are collected, and those specific parasites are taken here. They were washed and lysed by lysis buffer and tried to check what all proteins might be present inside the uh, parasite after this drug treatment, basically. So how much it is affecting the parasite basically because of these drugs or combination of drugs. So when they found that using this drug alone, few of them were upregulated, like membrane-associated calcium-binding protein, heat shock proteins. These were upregulated with this drug, whether they were also upregulated with the other drug. Whereas the other kinase, aldolases, enolases, etc., were downregulated with this drug, whereas few were upregulated with the other drug. When combinations were given, stress response protein were upregulated and glycolytic enzymes were downregulated. So they found that alteration of the parasite proteome induced by two components of these two drugs, and they basically established a relationship between pharmacological relevant concentration and time of exposure for these two components, and they found that how much it is affecting the parasite and how. So basically, that study will help you all to design the treatment strategy and help better treatment of to kill the specific parasites and get a person would be cured from malaria accordingly. So that's the application of 2D gel electrophoresis. Similarly, SARS analysis were done in one of the study. So this is how SARS virus looks like. It has phosphoprotein, it has glycoprotein, spike proteins, etc. So when they tried to study the healthy samples versus healthy patient versus SARS patients, and they did the whole protocol of two-dimensional gel electrophoresis, and they found that few of the proteins were upregulated in healthy control, where few were upregulated in down in SARS patients. So basically, what we can do is we can take this specific protein, cut this specific protein from the gel, and subject it to mass spectrometry, wherein you can understand what type of protein it is. So in this specific technique, you can just say what might be the isoelectric point of that protein and what might be the molecular weight of the protein, but you don't know what protein it is. You can only talk about that by using mass spectrometry. So wherein your proteins are fragmented after trypsin digestion. So you make different peptides, and that peptides can tell you, those unique peptides responsible can tell you about what this specific protein is. So MS1 and MS2. So MS1 is the initial fragmentation, and for example, if your protein is around 200 kilodalton in mass spec in MS1, it will get fragmented into, for example, 120 and 80. Then in mass spec 2, that's 120 fragment will further get fragmented into, for example, 120, for example. So that's what help you all to understand the exact protein which might be present in the sample, and it will help you all to understand the type of protein which might be present, which is upregulated and downregulated by comparing with the libraries by looking at the fragmentation pattern. So this specific indication you will be getting after doing this such kind of analysis. And there are few challenges also related to two-dimensional gel electrophoresis. This is how a good gel looks like if everything goes well. Similarly, this is another example of a good gel. But there can be something bad can happen by a number of different reasons. Something like gel to gel variation can be seen because of the samples, because of the electrophoresis technique you are using, because of the staining technique you used, while analyzing the way you have defined the spot boundaries, or there can be user bias as well. One is analyzing in some way, another is analyzing in some way, and because of that, you end up getting a little bit different results, etc. Or else because of biological variations itself. Sometimes you get a protein something like this. This might be because of trace impurities. You will get streaking like this. So the buffer, which is made up of trace, has some impurities and making your gel look something like this. If you have used old timid while making the SGS gel, it might look something like this. If you have used crude sample itself, if you are not fractionated it, removed the highly dense protein, etc., a protein gel might look like this. Similarly, if you have used solubilization without precipitation, your gel might look like this. Similarly, while after first dimension, if you have just equilibrated with DTT and not with alkylating agent, your gel might again look like some streaking effect, something like this. And while visualizing, if you are not focusing it properly or over focusing it, your gel might not look that accurately. There can be variations between the individuals, and because of that, your samples might get affected. It might, so these effects can't be controlled basically, inherent one, or because of induced, because of biological, between biological states itself, the way you are taking out the sample effects can be. So these are the some which can be controlled, because you need to accordingly take at the same time point, same ethnicity or same state. All of those need to be considered while doing the sample preparation, by collecting the samples. So sources of variations can be because of the sample preparation, can be because of the technique which you are using, like during the IPD strip, whether the pH ranges were different. The Because of the presence of salt, the gel image might not look properly. The way we have run your samples, whether in low voltage, high voltage, if that is changed, then your proteins will also look something different on the gel. Equilibration time point, the so all those procedural changes, manual errors can lead to change in your gels. Those are the few challenges associated with two-dimensional gel electrophoresis. So that's what I just talked about. So I need to repeat a bit because I got locked out. So maybe we'll take a break of 5 minutes or so, and we'll do question and answers. Okay. Until then, if you are having any kind of doubts, you can just write the message for the same. Yes, any doubts? Yes, people. Okay, then I think we can start with Q&A. So the question says in 2D experiment, the equilibration step involves what? Yes, I want everyone to answer. Okay, yeah. A is the answer, reduction and then alkylation, obviously. So what does reduction do exactly? And what does alkylation will do? What reducing agent do we use? Yes, yeah, what does it do? And what alkylating agent do we use? And what does alkylation does? Yes, what do they do exactly? And why do we need to do that equilibration step? You can unmute and answer as well, like if not typing. Yep, it breaks disulfide bonds and iodoacetamide does what? Yes, It's basically alkylates the sulfides itself so that it doesn't get it should not form the disulfide bonds again. So which of the following prevent reformation? Yes. Which of the following component leads to clogging of immobilized pH strip and could not form complexes with proteins by electrostatic interaction? Yes. Nucleic acids? Is it only nucleic acid? Everything will be responsible for that, right? So it will clog the gels and it will change the pH of the protein itself and it will not allow isoelectric point to get achieved, etc. Everything will be responsible for that. So you want to detect phosphorhydrated proteins, which type of stain you need to use? Yes. So now we have used Tizol method, which we did in our last lecture. 720 micrograms of protein from sample. She has to run 2 DE using 18 centimeter IPG strip and further wants to identify proteins using mass spec. Select the closest amount of protein she should be using to start 2D. Okay, when I was explaining, I just talked about microliters, but along with that, there was micrograms also written in that slide. So, how much amount of protein she need to use? If the strip is 18 centimeter, it's 300 to 1000 microgram. Which of the following pH strips would you use if your sample contains proteins with acidic pI values? Yes. So acidic pI values indicating at the acidic pH, proteins will stop moving, like it will achieve its pI values in the acidic range. So which strip would you use? Which has 7 to 10, 3 to 6, or 3 to 10, or 5 to 7? Yeah. Rohan, when visualized his 2DE, found vertical streaks. Which of the following factor may cause the streaking in the gel? Yes. No idea. Yeah. So in a gel, there are two proteins with same isoelectric point, but they differ molecular weight. What will be the position of this protein spot on gel with respect to each other? At same position, they will be overlap adjacent to each other or vertical to each other. Good. So here are two proteins with two separate 2D databases. Now you want to study and compare differentially expressed proteins. Which of the following software can be used? Oh, we didn't do this exactly, but just give a random guess though. Yeah. So that's basically a software which helps us to compare and study differentially expressed proteins. So which of the following is the correct sequence of events followed by serum proteomic studies? Yes. Yeah. So first tourniquet lies the cell. All the proteins will be coming out. Whatever is the high abundant protein, you will deplete them or remove them out first, then precipitate the protein and then quantify it. Yeah. Which of the following is not correct for 2DE rehydration process? Not correct. What happens in rehydration process? Not happens basically. Not correct. Yes. In 2DE, the answer is salts are not removed in the process. It's not done during that stage. It's during the sample preparation itself. In 2DE, the component of gel which maintains the pH gradient are called as. Oh, if you know it, you know it, but we didn't do in the lecture though. The one which maintains pH gradient is, yeah, yeah, ampholytes, the way by which those pH strips are made. In 2DE, second dimensional electrolysis is done at dash degree angle related to first dimension. One is like this, one is like this. Yes, which angle? Like one runs like this, another runs like this. So what is the angle which is made? In analysis software, which parameter is used to compare gels on spot by spot basis? So, Yes, it's spot analysis when you're doing spot by spot basis analysis. So although it's slightly confusing though, but the answer is spot analysis. Which of the following staining dye is not compatible with mass spec? So it's the silver stain which is not compatible exactly. So in silver staining, how is the reaction stopped? By adding acetic acid. Yeah. Which of the statement is correct about IEF? All of the above. So it does dissolve the protein on the basis of pI. It involves pH gradient shift and presence of salt interferes the process. So now the questions are lab-based questions. So which of the following statement about image quantile software is true? Yes, it's all of the above. So it can remove the background. It can be used for quantitative proteomics and it can employ standards for molecular weight studies as well. So I didn't cover this session though. There was one of the professor who talked about this software in detail. So which method is preferred for 1D gel analysis of complex proteins? Yes, So it's stepwise, which is generally used for one-dimensional analysis. Which of the following analysis cannot be performed by the software? Colony counting, array analysis, Dij analysis, or 1D general analysis. So you all will able to answer these questions only if you have gone through that lecture by one of the professor who talked about these softwares. So this is a different technique itself, wherein different dyes are used. So this, which we'll be learning in our next lecture about dyes. So this software cannot do dyes analysis, whereas all others can be done. So you had run SDS page and you were doing analysis using the software and wrongly selected one of the parameter which did not give you desired results. What is that option in software? Differentially express proteins from serum. So it's the fixed the bandwidth and that will help you all to that is something which because of which it did not give you desired results. If you have selected automatic band detection, it would have helped in analyzing the results. So all these questions from 16 to, I mean, from 17 to 20. So that's about this specific software, and one of the professor has talked about this software in detail in the session. You all can go through that, yeah, about that software. So I don't have that software, so I didn't talk about it much. Okay. Anything else you all wanted to ask or talk about? Yes. Yeah, tell me, anyone, if you wanted to ask anything else. Alpana, Anish, Himanish, Shivanjali. Shivanjali, I didn't see you like giving any answers or asking anything. And Veena, yes, anything you all wanted to ask about, talk about, nothing as such. If not, then maybe we can end the lecture. If you have any doubts, you can ask me right away, or else we can end the lecture here. Yes, Okay. Okay, thank you then for attending the lecture. We'll meet one more time next week, and as this is a 4 week course, and that would be last lecture. If you all want me to take one quick revision lecture of all 4 weeks, I can take one extra lecture, maybe next to next week, if they allow. I like they generally send an email if we need one more lecture. So if you all are interested in that, maybe I can take one lecture wherein I'll just brief about each and every whole 4 week lectures. Or else we can just do question and answers about different question and answers about 4 weeks. If you all need that, we can do that. Not next week will be the 4th week lecture, and then in the 5th week or 6th week, they basically ask us if you want any one extra lecture, we can do that maybe for an hour or 2 hours, depends. Okay, then thank you for attending this lecture. You all can let me know in the next week as well if you need that, then we can accordingly do it. Thank you.